Hello, this is Ed McNabb and Tom Hsiang presenting the progress of the project Practical Detection of Fungicide Resistance in the Field as of August 2021. The goal of this project is to develop a field kit for turf managers to determine if isolates of the dollar spot fungus from their turf have developed a reduction in sensitivity towards DMI fungicides. This kit will involve turf managers collecting and placing grass blades with symptoms of dollar spot onto a prepared growth medium. This medium would then be incubated for a few days, and the level of fungal growth can then be used to determine the sensitivity of the fungal isolate to the fungicide. We'll start with a brief background and some justification for this project. In 1979, DMI fungicides were registered for use against turf diseases in the US. Within 10 years of registration, reports of the fungi developing resistance to this family of fungicides occurred. In Canada, the first registration of DMI fungicides against turf diseases was in 1994. The Hsiang Lab has been monitoring resistance to DMI fungicides in dollar spot ever since. The research around this pathogen fungicide system suggests that repeated use of a DMI in an area can result in populations of a target fungus with reduced sensitivity towards a used fungicide. We arrive at the problem we seek to address with this project. Normally, assessing the sensitivity of a fungal isolate to a fungicide requires time and human labor. We are seeking to reduce these along with the technical requirements for such tests. A simple field kit would allow turf managers to sample leaf blades displaying symptoms of dollar spot and assess the level of resistance of the fungus to a fungicide. Funding from the OTRF and research for this project began in fall of 2020. Ed McNabb was taken on to pursue this project as part of his master's work. By the end of the fall of 2020, we determined a preliminary discriminatory medium that is capable of producing a visual difference under lab conditions between pure isolates of the dollar spot fungus that are sensitive to DMIs and isolates that are resistant to DMIs. In winter of 2021, we tested the discriminatory medium with turfgrass samples grown and inoculated with the dollar spot fungus under controlled conditions. This test showed that the discriminatory medium could differentiate between sensitive and resistant isolates of the dollar spot fungus in infected leaf blades. In the summer of 2021, we began testing the field kit with samples of turfgrass from the Guelph Turfgrass Institute. Here are the original objectives from the proposal. First was to develop a preliminary discriminatory medium for use with lab fungal cultures. This was completed in the fall of 2020. Objective 2 involved using the discriminatory medium with infected leaf blades. This is currently ongoing. The third and fourth objectives are to come, and they involve asking turf managers to test the field kit and potentially extending this test to other diseases. The first objective was completed in the fall of 2020. In short, we began by reviving several isolates of the dollar spot fungus with varying sensitivities to DMIs. We then prepared an array of growth media that was amended with increasing concentrations of propiconazole to determine a concentration of propiconazole that inhibited the growth of sensitive isolates of the dollar spot fungus while allowing resistant isolates to grow. From this test, we determined that media amended with propiconazole to concentrations between 0.5 and 1.0 micrograms per milliliter was potent enough to inhibit sensitive isolates of the dollar spot fungus, but not too potent as to stop growth from resistant isolates. This concentration of propiconazole, along with antibiotics or a weak acid to prevent some bacterial growth, is what we consider to be the preliminary discriminatory medium. Once a preliminary discriminatory medium was determined, we proceeded to tests with turfgrass samples. During the winter of 2021, we seeded containers with creeping bentgrass and grew them in the lab. This was to minimize external contamination for initial testing. Once grown, Each container was inoculated with an isolate of the dollar spot fungus with a known sensitivity to propiconazole. Once symptoms of dollar spot were observed on the leaf blades were sampled and placed on the discriminatory medium for testing. The image on the right shows the results of these tests and illustrates the capacity of the discriminatory medium to differentiate between DMI-resistant and DMI-sensitive isolates of the dollar spot fungus from leaf blades within 96 hours. Once the discriminatory medium was shown to work with leaf blades inoculated with the dollar spot fungus, we could then proceed to testing this medium with field samples. Before environmental conditions were suitable for dollar spot infection in the field, we began assessing leaf blades from turf at the GTI for organisms that may interfere with the field kit. Starting in March of 2021, we began sampling turfgrass leaf blades from 4 greens at the GTI on a bi-weekly schedule. These samples were placed on the discriminatory medium and monitored over the following week. Any organism that was able to grow on the discriminatory medium within 1 week was assigned a morphotype and prepared for future identification. Filamentous fungi and yeasts that were able to grow on the discriminatory medium within 96 hours and resemble or obscure the fungal growth of the dollar spot fungus were of particular concern. Such fungi may affect the interpretation of the results of the field assay and will need to be isolated, identified, and inhibited before future distributions. Once environmental conditions became suitable for the dollar spot fungus to infect turfgrass, field trials at the GTI were prepared. Once at the GTI, greens for the field trials were selected and inoculation plans prepared. The inoculation plans involved 6 isolates of the dollar spot fungus, 3 of them sensitive to DMIs and 3 of them resistant to DMIs. A grid of 24 inoculation points allowed for 4 replications of each isolate in the field trial. The space between the inoculated areas provided a buffer region to minimize the spread of the fungus between the inoculated areas. Inoculation plans were outlined using a fiberglass tape measure, golf tees, and fluorescent spray paint. The tape measure was first used to outline the perimeter of the trial area, and golf tees were planted in each of the corners and at each 1-meter vertex to create the grid. Each golf tee was then highlighted with a spot of fluorescent spray paint for future reference. Once each 1 meter square was clearly defined, the prepared seed inoculum for the dollar spot fungus was spread in the center 0.25 meters square of each 1 meter block using a simple template. Inoculated plots were then irrigated to assist with disease establishment and monitored over the following weeks. Once dollar spot symptoms were observed, sampling and testing of the leaf blades with the discriminatory medium began. Grass blades were sampled from replications of each isolate during the field trials. Isolates 1 to 3 are considered sensitive to DMIs, and isolates 4 to 6 are considered resistant. From our previous test, we would expect to see fluffy white growth consistent with that of the dollar spot fungus on isolates 4 to 6, with no such growth or very minimal growth on isolates 1 to 3, as shown in these images. However, the white fluffy growth is much more apparent on the growth medium on the right. This represents the media amended with 0.5 micrograms per milliliter of propiconazole. This suggests that this lower concentration may be better suited for use with field samples, as it makes the results much more apparent. One point of concern is the presence of the yeast colonies growing on these field samples. This may be obscuring or reducing some of the fungal growth of the dollar spot fungus. In addition to these yeast colonies, we have also encountered some filamentous fungi that may be of concern for this field assay. Both these yeasts and filamentous fungi may interfere with the way we interpret the results of this field assay. Currently, we are working to reduce the growth of the yeast colonies and other non-target filamentous fungi on the field kit before we distribute it to turf managers. Once results of the field kit are consistent and easily interpretable, turf managers across Ontario will be contacted for participation in the initial distribution. Turf managers will receive the field assay via Canada Post, along with a short instructional booklet and a way to report the results and any complications. This information will allow us to revise the kit and prepare for additional distributions as well as a final revision. Once suitable performance of the field kit is achieved, the proof of concept can be used to develop similar kits for other pathogen fungicide systems. Other pathogenic fungi that display rapid growth on standard growth media and have easily identifiable characteristics, such as the fungus responsible for gray mold of strawberries, would make excellent candidates for similar field kits. Thank you for your time and consideration.